Hello ladies and gentlemen, the title of this presentation is Protein Separation Techniques. The purification of proteins is an essential step in understanding their function. The isolation and purification of a single protein from cells containing a mixture of thousands of unrelated proteins is achievable through several steps. 1. Extraction from biological material. 2. Precipitation, initially to recover the bulk protein from a crude extract. 3. Use of Size-Exclusion column and then a lon-exchange chromatography to further separate the target protein containing fraction from the bulk protein. 4. Using the Affinity chromatography to enable the recovery of a target protein in a highly purified state along with a high yield. Now we will start explaining each step aside. The first step is to release protein from cells and tissues by homogenization. The membrane of tissues or cells is disrupted by using a blender or a sonicator, which is an apparatus that releases sound waves to open cells, or by using a detergent if the proteins are tightly bound to other biomolecules. This step has to be done at 4 degrees to minimize the denaturation of proteins. Then you have to centrifuge the mixture. The next step is to choose the separation techniques, knowing that each one depends on a specific property such as solubility, size, charge, and natural interaction. For example, the salting in and out technique depends on the solubility of protein, which in turn depends on the concentration of salt. The ammonium sulfate is the most common salt used because it's cheap and it's harmless to protein. At low concentration of salt, the solubility of proteins increase. This is known as the salting in. At a certain salt concentration, the proteins reach their maximum solubility. At high concentration of salt, the solubility of proteins decrease. The proteins tend to associate with each other and precipitate. This is the salting out. In order to separate the target protein, we must add enough salt to precipitate it. Then, we centrifuge the solution to collect the precipitate. The next step starts by dissolving the precipitate containing the target protein in a buffer and dialyze it to remove the salt. The dialysis of the ultrafiltration separates protein on the basis of size. The dialysis technique is used to separate the target protein from small solutes like salt by diffusing through a semi-permeable membrane. So, the solution of protein is put in a bag of cellulose membrane having small pores of controlled size. Proteins bigger than the pores are retained while the smaller molecules may diffuse out. This technique is usually used to remove the ammonium sulfate so it won't interfere while using the ion exchange chromatography. The ultrafiltration technique uses pressure to force a solution through a semi-permeable membrane of defined pore size. The solvent and small molecules having low molecular weight pass through the membrane, forming the filtrate, while high molecular weight remain in the retentate. This technique serves for the purification and the concentration of protein. Next, we will talk about the gel-filtration chromatography, which is a separation technique based on size. Size-exclusion chromatography, also known as gel-filtration chromatography, depends on the differential migration of dissolved solutes through gels. A mixture of molecules of different sizes is applied to a gel that consists of beads of polymers such as agarose with defined pore size. The small molecules enter the pores of the gel while the larger ones migrate and elevate. The smaller molecules eventually elevate out of the column by changing the pH or the concentration of salt. This technique is frequently used for protein purification and for desalting protein solutions. After the removal of salt and small molecules by dialysis and gel filtration chromatography, we use the ion exchange chromatography. This technique uses a charge on a protein to separate it from other proteins. So the proteins, depending on their net charge, can bind either to cation or anion exchangers. The arm of cation exchanger is usually the carboxymethyl cellulose, while that of anion exchanger is the TAE arm. For example, at pH below the isoelectric point, the amino acids of proteins are positively charged and will bind to a negatively charged resin called cation exchanger resin having carboxymethyl cellulose arm. Finally, the protein can be eluted from the column either by changing the pH or by increasing the salt concentration, which shields the charges and thus decreases their attraction. The affinity chromatography is another powerful and generally applicable means of purifying proteins. It's based on the high affinity of many proteins for specific chemical groups. This technique is based on the natural interactions between a protein and the ligand. The polymer forming the tertiary phase is covalently linked to a ligand that binds specifically to the desired protein. The elution of the bound protein could be by using one of the three methods, either by adding a high concentration of a soluble ligand, or by competition with a bound ligand, or by changing the pH to disrupt the protein-ligand interaction. This technique is convenient and the products are very pure. It's used for antigen-antibody, enzymes, inhibitors, and others. Thank you for your attention. If you find this video helpful, please like and subscribe.